Good evening, everybody who's joining us this evening. Welcome to the British Society for Immunology's webinar on how effective are COVID vaccines for people with weakened immune systems. I'm Doug Brown, I'm Chief Executive of the British Society for Immunology, and it's my absolute pleasure to be hosting and chairing the session this evening. Those of you that don't know about the British Society for Immunology, in a nutshell, we are a professional society and a charity. We represent all things immunology, and we have thousands of members across the UK and across the world who are working within the immunology space. We've had, as you can probably imagine, a really big role during the pandemic, given the proximity of immunology to COVID-19. And we've been working with the research community to help researchers coordinate and bring their research together as best they can so that we can understand COVID-19 as quickly as possible. We've been working with policymakers to bring the experts to the policymakers to inform and advise on policy decisions. And by no means least, we've also been developing a lot of public facing information that's available on our website about the virus, about COVID-19 and about vaccines as well. So if you haven't already, please do visit our website. There's loads of information on there and it's all free to access. We will post a link later on in the session here, and we'll send a link out via the email after this session too. So please do have a look. But you're not here to listen to me talking about the British Society for Immunology. You're here for this webinar, and I can guarantee you are in for a treat this evening. We've got an amazing session lined up for you this evening, and we've got hundreds of you logged in viewing this live, which is absolutely fantastic to see. Firstly, we're going to hear from Professor Sir Jeremy Farrar, who is the director of the Wellcome Trust and involved in various government bodies and has been very busy during the COVID pandemic. And he's going to be setting the scene, talking about the pandemic from UK global perspective, but also the monumental research effort that has happened over the last 18 months or so. Then we'll hear from Lynn Laball, who is a patient representative involved in supporting research and has been for many years, but absolutely crucial during the pandemic. And she will be bringing her patient perspective on the pandemic and the research that has been happening. And then we have Dr. Michelle Wellcome and Professor Paul Moss, who are on the front line of delivering that research. Lots of clinical studies and lab-based studies that have been conducted over the last 18 months. They've been there leading many of them, and they're going to tell us about their most recent findings in relation to vaccines and people with weakened immune systems. So we got loads of great stuff that will be coming through from our speakers, and it's going to be interactive as well. So we have a Q&A session at the end. Hopefully we'll have 20 to 30 minutes available for that. So please do post your questions via the Zoom question link. I have a trusty assistant who is going to send me the questions via WhatsApp. So I will ask as many of those questions as possible of our speakers this evening. Of course, and you'll understand, we can't answer any individual medical questions, but questions generally about this topic will be gratefully received, and I'll ask as many of those as possible. So without further ado, I'm going to hand over to our first speaker this evening. This is Professor Sir Jeremy Farrar. You may have seen him on the telly. He's been very busy over the last 18 months, but as I said, he's the director of the Wellcome Trust, which is a huge charity funding many millions of pounds worth of medical research every year. Had a great key role within the pandemic over the last 18 months. He's also a member of the government SAGE committee and the government's vaccine task force and various other bodies based in the UK and overseas when it comes to the pandemic. So as I say, without further ado, Jeremy, I'm going to hand over to you to set the scene for this evening, please.
Doug, thanks very much indeed, and I hope everyone can hear and even see me. But just start to thank the British Society of Immunology, Doug, and you and your whole team for suggesting this webinar in the evening. And I hope it's of real value and interest and to everybody that's been able to join this evening. It's a great pleasure to be able to join with Lynn and Michelle and Paul on this webinar. Yeah, so my background is in infections and emerging infectious diseases, and I first became involved in this pandemic on December the 30th or 31st of 2019, and even then, nobody could have predicted what has happened since. And here we are in September 2021, and the pandemic is still with us. And the truth, at least in my view, is that the pandemic won't suddenly come to an end. It is now shifting into a new phase. I think we've witnessed a new human infection emerging at some point in December 2019, and now being part of humanity, and I think part of humanity from now on. And that requires, I think, a new way of thinking. Over the last 18 months, 20 months, of the most remarkable things has been the advance of science, of which many have played a role, including everybody who's joining this call tonight. The scientists themselves around the world, but also the volunteers and the people that participated in studies around the world, and everybody's thanks should go out to them, because without them, the progress in the scientific endeavor would not have happened. And perhaps the most remarkable advance has been the development of vaccines. The UK in particular would be in an absolutely awful position today in the absence of having incredibly safe and incredibly effective vaccines. But vaccines don't work in public health on their own. They work with everything else. They work with testing, they work with behavior change to some degree, and they work with treatments for people that do inevitably get ill. And of course, vaccines may not work for everybody, even though we say they're incredibly safe and they're incredibly effective, 95% effective against severe illness. That still leaves a group of people who are not protected from the vaccines. And that's really the focus of this evening. Those people who may worry if they are protected, those people who may not be protected, and those people certainly who are not protected. There is also a lot of information out there in the media, some of it very good and very accurate, and some of it less accurate and less good, and some of it downright dangerous in my view. And I hope this evening goes some way to bringing some sense to all of the information, whether it be on boosters, whether it be on which vaccines to have, the mixing, maxing the vaccines, how long they last, and who they work in. And if they don't work, what can we do to help people to be protected from what remains a very nasty infection indeed? So, Doug, I just wanted to set the scene for a few minutes on where I think we are in the pandemic and the role that science has played, and then hand back to you, to Lynn, to Michelle, and to Paul to go through more of the detail, and then opening up for questions at the end.
Fantastic. Thank you very much, Jeremy. Such a fascinating insight from somebody who's been so important in bringing together the evidence, advising government, and really pushing things forward. So thank you very much, Jeremy. And as you say, I'm sure there's going to be lots of questions, and we will come back to the whole panel in due course. So it's with my absolute pleasure to be introducing our next speaker, who is Lynn Laball. She is an experienced patient public representative on research projects, and has been for many years, well before the pandemic. But we had the absolute pleasure of working with her on a really important program of work that you'll hear more about from Lynn, but also Michelle and Paul, called the UK Coronavirus Immunology Consortium. And Lynn was at the core of that, with a group of patient representatives really shaping what should be focused on in that program. I'm sure Lynn will tell you lots about that, but she works with many other charities, many universities and other bodies as well. So absolute expert in this space. And Lynn, I can't wait to hear your patient perspective on the pandemic and the research that's happened. So Lynn, over to you.
Hi, thanks, Doug. I'm not sure where to go with such a lovely introduction like that. So, hi everyone, my name's Lynn Laball, and I live with a rare autoimmune rheumatological disease, a form of inflammatory arthritis. And like many of us, once you get one thing, there's a whole load of things come along, like the number 9 bust, isn't it? So I have multiple long-term conditions, including problems with my heart. I'm immunosuppressed. I was classed as extremely clinically vulnerable and had to shield. I'm on a biologic medication, an anti-TNF, and I take methotrexate as well. And I realised that people will have different concerns in relation to their personal circumstances. And I'm not here to be representative of all patients, but I suspect that many of us share common concerns and frustrations. And some of the things I'm going to say is a personal point of view, and I don't want my comments to be viewed as criticism of researchers or clinicians, because I think the past nearly up to 2 years now has been really challenging for all of us, hasn't it? And I was involved in the COVID Immunology Consortium Patient and Public Involvement Group. And for those of you that don't know what patient public involvement is, it's been described as research that is done with or by the public, not to, about, or for them. And as Doug says, I'm involved in multiple different groups and research projects across the UK, including, and one of the, well, a great personal thing for me is that myself and another patient and public contributor had a research idea, wrote a grant application, and we've been funded to look at the views of people who live with arthritis on what happened with shielding, with a view to, you know, if we were ever in this situation again, what would be really important to people that are shielding, and how would they like to inform that process. So I thought long and hard about what I wanted to say, and it's been really challenging for me to get my thoughts into order. And I realised that actually what I really wanted to talk about was the emotional effects of the past 2 years, kind of seen through the lens of the experiences with my conditions and also with involvement. And I'm involved because of my lived experience. I had a 4-year diagnostic odyssey because I have a rare disease, and I used all the research that I could access to help me on my journey. And to me, research is hope. And I think that no one will ever want research to succeed more than people living with conditions or people that are part of the communities affected. And I think that's been one thing about the pandemic, hasn't it? There's been an absolute explosion of interest in research and a real understanding of what research means and the importance of what we're doing having an evidence base. So, and in my experience, that's what drives people to be involved in research, whether it's to be involved with researchers or whether it's to participate in research. That need to make research better, to make it more responsible to people, to cut research waste, and to help, even if what we're doing won't help us at this moment in time. There's a real emotional connection there. And at the start of the pandemic, patient and public involvement kind of disappeared. And in many ways, it was seen as too time-consuming in this first fast-paced environment that we were in, that, you know, we had to get research, and we had to get it quickly. And I wonder if in reality it was never as embedded as we thought it was. And then things picked up. And I got involved in the COVID Immunology Consortium, which has just been a fantastic experience. I think the British Society for Immunology and Erica and Doug and all their colleagues worked really hard to put together a group of people that were representative or represented, you know, everyone that was affected from COVID, whether that was because you belonged to a certain ethnic community, whether it's because you were immunosuppressed, and lots of other things. And, you know, we really used our lived experience to challenge the researchers and their assumptions. But I think our biggest success was the way that we helped disseminate the research outputs in an accessible way. So we helped to write plain summaries, we did videos, we inputted on infographics, and we helped in events like these. And as Doug said, there is a wealth of really wonderful information on the British Society of Immunology website. And, you know, I think we felt a part of the team, that researchers came and spoke to us and presented their research and really listened. And the feedback that we got was that it kick-started them thinking about things, that our perspective got them thinking maybe in different ways and thinking about research that they needed to do in the future. And I think that the communication has been all important. because it hasn't been great for many of us in this pandemic. Personally, I was missed off the shielding list. I think that a lot of people have felt there's been a lack of information and we haven't been able to access resources. And I think a big challenge for a lot of us has been the lack of personal information for us, as opposed to this generic information that's been put out. Because we want to understand our personal risk. We want people to understand our personal circumstances to enable us to make decisions, to do the right thing, to protect ourselves. And this leads me to kind of what I hope this webinar will concentrate on, and I think it will. And this is information about the COVID vaccine and immunosuppression. So I've read the preprint of the Octave study, which looks at the vaccine. The vaccine was given to certain groups that were either immunosuppressed or lived with renal disease or liver disease or cancer. And I checked out their website, and they did have some patient and public involvement. But I'm wondering if they could maybe have done a bit more to help disseminate their findings in a way that made sense to people, because I think a lot of us have been asked with a lot of questions. I think there's some confusion about, the protection that we have from the vaccine, whether it's protected us on a cellular level, whether it's the antibodies in the blood that are important. I know the JVCI has announced third doses for certain people, and they've said that this will be clinician-led, but I'm not clear what the process is. I know that I need a third dose, but it looks like I need to wait until I'm identified as needing a third dose. I can't identify myself, and it feels like I have to be a passive recipient of a process again. So it feels uncomfortable to me being defined by our vulnerability when we try not to be defined by our disease. And going forward, I think we need better, more personalised information, improved communication, and people working alongside research teams from the start. The big success of COVID has been the way that communities have come together to solve problems and find solutions that work for us. And what I would say to researchers, to clinicians, to everyone is involve us, let us help. Thank you.
Fantastic. Thank you so much, Lynn. Such an amazing and inspiring perspective there. And again, I'm sure there'll be lots of questions for you when we come to the Q&A. So please do, everybody, keep bringing that, putting those questions in. We are going to ask as many of those as possible of our speakers. But before we get there, just to introduce our two final speakers who are going to do a double act for the next 15 minutes. We've got Dr. Michelle Wellcome, who's a senior clinical lecturer and honorary consultant nephrologist at Imperial Healthcare NHS Trust. And following Michelle will be Professor Paul Moss, who's Professor of Haematology and Deputy Head of the College of Medical and Dental Sciences at the University of Birmingham and the lead researcher on the UK Coronavirus Immunology Consortium. So over to you, Michelle and Paul, and tell us about what you found recently with regards to immunity and COVID.
Thanks very much, Doug. Can you see my slide?
Yes, we can.
Yeah. Brilliant. Okay, good evening, everybody. So I'm going to present some data on the immune responses to COVID vaccines in immunosuppressed patients. If I can advance my slides. We know that in immunocompromised patient populations, when they catch COVID infection, they are at higher risk of severe disease. And typically, we also know in this patient group that they have inferior immune responses to common vaccines and were excluded from the initial COVID vaccine trials. And therefore, at the time of clinical rollout, there was no data on the immunogenicity or the efficacy of the vaccines in these patients. So this slide summarises studies which have reported seroconversion or antibody detection following COVID vaccine across different immunosuppressed patient groups. And as you can see, there's a lot of variability between each of the groups, but there's also a huge amount of variability within each group, which is confusing and complicated when we're trying to provide estimates for patients. And there's lots of different reasons why there may be some variations. Some general considerations are what vaccine type people have received, and certainly most of the data that has been published is on the mRNA vaccines. The dosing interval, for which the UK is an outlier, which test and what assay is being used to test for the antibodies, and the timing of the test post vaccination, because we do know that antibodies do wane over time. And there's other considerations such as age, as we know that younger people tend to make a better immune response or a response to the vaccine. But for immunosuppressed populations, there are specific things which may influence whether or not somebody develops an antibody response or immune response to the vaccine. So you need to consider the diagnosis, so what the underlying pathology is and whether or not the disease is active. We also must consider the treatment, so what someone's on at the time that they have a vaccine, and also past treatment may influence vaccine responses. And importantly, the timing of treatment with vaccination all may influence immune responses. And a lot of this variation is circumvented in the OCTAVE study, as Lynn has already highlighted. So this is a multi-center study which is looking at antibodies, T cells, and vaccine efficacy across different immunosuppressed patient cohorts, all using the same protocol, which will provide us insights unlike the individual studies that have been published. I'm pleased to say that the OCTAVE study sort of preliminary interim results were published last month, and at the time over 2,500 patients had been recruited. The report was listed 455 patients, and 89% of patients had antibody. But once again, we saw a range of antibody responses amongst the different groups, and so only 27% in some of the patients receiving active treatment, compared with 100% of patients in the inflammatory bowel disease cohort. But of note, 4 in every 10 patients did have an antibody level that was slightly lower than the healthy controls. But reassuringly, there were comparable T cell responses across the different cohorts. So I'm going to move on to talk about immune responses in specific patient groups. And I'm a transplant nephrologist, so I look after patients with end-stage kidney disease. And infection, sadly, is one of the leading causes of death in patients with end-stage kidney disease who are immunocompromised. They're immunocompromised on dialysis due to underlying disease, and with transplants due to the need for lifelong anti-rejection medication. And together with the OCTAVE study team, the team at Imperial have investigated immune responses to vaccine in 1,900 patients with end-stage kidney disease. So first of all, the results of 1,000 dialysis patients. Firstly, to say that when we investigated, we found that 45% of these patients actually had evidence of prior infection. And I think it's important to highlight that immunosuppressed patients have needed to continue treatment, life-saving treatment throughout the whole pandemic, and certainly through the first wave and the second wave, they were particularly affected with high infection rates. But for those patients who didn't have prior infection, 86% of them developed new antibodies following vaccination, with no difference between Pfizer and AstraZeneca. And 86% compared with other vaccines is good. But the antibody levels in those patients with no previous infection, once again, were lower than those who were vaccinated following a prior infection. So next we looked at kidney transplant patients, and we found that 16.5% of our patient cohort, and we're based in West London, had evidence of prior infection. Most of this was asymptomatic infection. These rates were lower, slightly lower than the sort of general population in London at the time, but actually significantly high for a patient population who were considered to be shielding. But for those patients who didn't have previous infection, only 55% of these patients developed antibodies, and all these transplant patients must be maintained on anti-rejection medicine to prevent rejection of their transplant. We did find that patients who had Pfizer had slightly better antibody response than those who received AstraZeneca. But because we had studied quite a large number, we were able to look at individual factors associated with antibody development. And we found that those patients who were older, or those patients who were vaccinated in the early post-transplant period where their level of immunosuppression was the highest, were less likely to develop antibodies, while those who received Pfizer and those receiving less immunosuppression, so maybe one type of anti-rejection tablet rather than 2 or 3, were more likely to develop antibodies. We also looked at T-cell responses, so T-cells required to fight infection, and the T-cell responses were quite low in the transplant patients, and this is not entirely surprising, as the anti-rejection medicine targets the T-cell activity. This is in contrast to patients who have autoimmune disease, who are often on treatment to prevent antibody development. And we looked at some renal patients who had autoimmune disease, and we found out of 91 patients, 59% of them developed antibodies, but even more of them, so 83%, developed T cells. And once again, we found that those who had had recent treatment with an agent that affects B cells called rituximab, or those with evidence of ongoing effect of rituximab, were less likely to seroconvert. And before I hand over to Paul, I just want to highlight the OCTAVE Duo trial. So the OCTAVE team have continued now onto a second trial called OCTAVE Duo, which is a randomized trial looking at the different vaccine strategies in the same patient population. So we're aiming to randomize 1,200 patients, again, all immunocompromised, to either receiving Pfizer or Moderna as a 3rd dose vaccine. And some patients with lymphoma will also receive a 3rd vaccine. And recruitment's going incredibly well, and we're almost halfway through. So we're hoping that it won't be too long before we can report in full in this trial. Okay, so I'll hand over to Paul.
Thanks, Michelle. That was super and great introduction for my work. Thank you very much. So I'm Paul Moss, as Doug said, from the University of Birmingham. And the next slide addresses my topic tonight, which is really just talking about patients with a cancer diagnosis. And obviously the cancer itself and its treatment can both impact on vaccine responses. This is a complex topic, and it depends on many factors, the type of cancer and the type of treatment. And of course, active chemotherapy, if you're on chemotherapy, it's likely to suppress antibody responses. That we know that drug is designed to stop cells from dividing. That's no great surprise. But several types of blood cancer, in particular, are associated with marked immune suppression. And I am a hematologist, so that leads me on to this topic that I want to talk to you about tonight, which we've led here from Birmingham. It's called the Chronic Lymphocytic Leukemia Vaccine Response Trial, or CLLVR. And on the left, you'll see a blood slide. All the hematologists like myself like to look at blood slides, and you'll see those purple cells are lymphocytes, and there's too many of them. That's why leukemia, white cells. And this year, we were able to recruit 500 patients across the UK and study their antibodies and the cells, antibodies in red and cellular immune responses we're just starting to do in green. And we've one published paper. And I just on the next slide there, I want to go back. I just want to thank people who've done this. Helen and Rachel and Graham have been remarkable. And it's funded by National Core Studies and great charity support. The UK has great charities. CLL Support Association, Leukemia Care, and Blood Cancer UK. And on the next slide, these are our results expressed in a similar way to Michelle. And after double vaccination, we are seeing antibody responses in this patient group, 67%. Whereas you can see the HD, the healthy control donors, it's 100%. So this group of patients, the nature of this leukemia makes patients immune suppressed. So only 67% respond. And even of the people who respond on the graph below, their antibody levels are 70% lower. So you can see that these results show that antibody responses in this chronic lymphocytic leukemia to vaccines are suppressed. And many of you perhaps on the call have contributed to CLLVR, and I'm very grateful for you. Believe it or not, we believe that in the last 9 months, this has become the biggest vaccine study ever in leukemia. And that's reassuring, but it's also a bit depressing for me that we haven't done more vaccine studies for this group of patients over the last few years in other areas like influenza, and we will carry that legacy forward. Now, the next slide shows the vaccines in our hands were the same. The Pfizer and the AstraZeneca, in those two pie charts, showed equivalent antibody responses at 66% exactly. On the right, you can actually see what we call virus neutralization data. In blue is the neutralization against the original virus from Wuhan, which actually is in the vaccine. That's the type of gene in the vaccine. The red is the Delta. All infections now in the UK, almost all are Delta in red. And if you look on the left, you'll see that red and blue look pretty similar. That's those dots. That's healthy controls out in the population, showing how well their blood neutralizes Wuhan or Delta. But just adjacent to it, you'll see a blue and a red, which are lower. And those show those from CLL patients, leukemia patients. And look at how that red drops right down. So our CLL patients are making poor antibody neutralization of Delta, the virus in the community. The next slide shows a nice little story, I think, on how the stage of disease affects your response. So I've tried to animate it, Michelle, with the next. So that red box shows patients with leukemia who are untreated, and you'll see that horizontal bar showing their antibody levels. So they've not had chemotherapy. But the next bar shows people who've had treatments, that little block of 3 in the red square, and you'll see how their antibodies after vaccination drop down. That's the treatment effect. But after the treatment, if they respond well, the next final red box show that the antibody responses bounce back. So when you're looking at a patient with cancer and their vaccine responses, then you have to consider the stage of their disease and the nature of their treatment. Let me just move on to the next. That animation, I think, is quite interesting because many patients with CLL have rituximab, which was referred to earlier by Lynn and Michelle, this antibody that's used in all sorts of diseases. And you'll see that the black bar there shows that if you've had, as a patient, rituximab within the last year, then only 24% developed an antibody response. Whereas if it was a year ago, since you had the treatment, you're getting a good response, 56%. So again, we're starting to get some idea of how long you need to recover from these therapies in order to generate antibody responses. Now, the next slide shows what we're doing. This information is important, but we need more to protect patients. And so, as Michelle said, we've managed to link into this wonderful Octave Duo trial, which is testing a third vaccine, a booster vaccine. And 50 patients from this CLLVR study have been recruited into the Octave Duo, and the information should be available within the next week or two. And of course, now we know that the booster vaccine is going to be used for immune suppressed patients across the UK anyway. And as Lynn said, the logistics of that need to be worked out. But for those people, those patients who absolutely make no antibodies, no matter how many times, then we might have to think about something such as injection of an antibody into them. And that's in the 20th of August, there was some positive data from America, and we await the protocol on that. But my final slide really shows about clinical protection. How well is the immune suppressed population responding clinically to these vaccines? And it's pretty good. We know, as you'll see at the top, vaccines offer good protection against infection. But what they've been brilliant at, and Jeremy mentioned this right at the start, is protecting against severe outcomes. That's what we want. We don't want hospitalizations. Of course, we don't want deaths. And here, these vaccines in the population at large protect around 90% or more. But Public Health England, just last week, you'll see the date, 9th of September, had this quote: protection against hospitalization remains high throughout the follow-up period and even within clinical risk groups, which is what we're talking about tonight. Vaccine effectiveness against hospitalization, you can see the numbers, 75 to 90 with AZ and over 90 with Pfizer. So as it stands, the clinical protection from vaccination remains robust for most people, even with immune suppression, but we'll talk about more in questions. And with the next slide, I will hand back to Doug for questions. Thank you.
Wonderful. Thank you so much, Michelle and Paul. Such insight there into some of the cutting edge studies that have been happening. So we've had lots of questions in. I can see the panel have rejoined, which is great. So I will ask as many of these questions of the panel. Please forgive me if I've not been able to ask your question by the time we get to 7 o'clock. The number of questions coming, we could be here for days. But the first set of questions that have come in are probably more for Michelle and Paul, looking at some of the specifics of the studies that you've been doing and others have been doing as well. So there's quite a lot of questions looking at, questioning T cells. So we know a lot about B cells and antibodies, but what do we know about T cells? Not just the activation of T cells, but how effective is that T cell protection, in particular in people who don't have an antibody response or can't have an antibody response in response to the vaccination. So Michelle, Paul, it'd be great to get your insights on that.
Yeah. I might.
Sorry. You go, Paul.
Okay, I'm going to start with an anecdote just for fun, because when I did my PhD, I won't tell you how long ago, there's a certain gentleman called Jeremy Farrar who was working in an adjacent lab. My PhD was on cellular immunity, T cells, with a guy called John Bell, who you often see in the newspapers. He's super Sir John Bell. And so I've worked on T cells for 30 years. T cells are incredibly important, I believe, in protection against COVID. And when I talk about, when we report 90% affection against death and so forth, I'm really increasingly confident that T cells are really critical there. I didn't talk about T cells in my study, and we're now doing that work. What we're finding is that T cell responses is actually, Michelle said in Octave, and we'll go to Michelle in a minute, were quite robust. And there are some papers only in the last week, I think somebody sent it me last night, that patients on rituximab have still quite strong, potentially even stronger than control groups, T cell responses. Slightly different because of the rituximab, but robust. So they're incredibly important, Doug. That's a great question. Over to Michelle.
Yeah, thanks, Paul. Yeah, we found in the Octave study that between all the groups, T cells were comparable, and also with healthy controls, which was incredibly important. We're still doing ongoing work with regards to correlating the T cells with what immunosuppression patients are on, but there is encouraging data from the Octave study so far.
Brilliant. That's great. Thank you, Michelle. And we'll start seeing some of that data coming through, won't we, in the next couple of weeks, which is wonderful. And just an aside, Paul, Jeremy, it looks like you're fresh out of the PhD lab. So I wouldn't have said it was 30 years, was it, Paul, you mentioned? But thank you. I mean, great answer, and definitely some more insights coming through in the coming weeks, but a brilliant question there. We've also got a number of questions coming in asking how effective do you think the booster will be for those who are seriously immunocompromised? So again, Michelle, Paul, some thoughts on that?
Okay, so we know certainly, sorry, I'm very kidney centric, but from the transplant world, there's France have been given third boosters or third doses of vaccine since April time. Israel and America are all ahead of us. So there is some data emerging from those countries. And the only caveat is that they're using 3 doses of the mRNA vaccine, which is not entirely applicable to our patients. But a lot of data suggesting that people who have a small antibody response will boost to a third dose of vaccine. But those patients who don't have any antibodies initially may not have any antibodies after a third dose.
Okay.
Yes, I would totally concur with that. I mean, certainly within CLL population, those people who have a response detectable, I think they will boost. In CLL, we have people who have very, very low antibodies, probably because of the nature of the disease, and we probably can't expect them really to make a response. It's a small group of patients, so there we might not see the response. But overall, Doug, we're confident and hopeful that we will see improvement.
Brilliant. Lynn, I see your hand is up. Yeah, over to you.
Yeah, so I just want to say really quickly, so before I got my first 2 doses of the COVID vaccine, so the American College of Rheumatology had brought out some advice, and because I'm on methotrexate, and because I know that I can stop methotrexate for a couple of weeks, I decided to withhold methotrexate to try and give myself the best chance to, and I'm not saying to people to do that, but I do think that, you know, if we're going to do these 3rd vaccine doses, then people need to work with their clinicians to work out what's the best thing for them. And that's something that I don't see happening. So if your disease is stable and you think that, you know, the best thing to do is to withhold some immunosuppression, or it's not, but we need some help to make these decisions, to give us the best chance that we possibly can, because we don't know what level of antibodies or whatever are protective. And, you know, to me, it's just all about optimizing, but in a way that makes sense to us and gives us a bit of control over what's happening as well.
Brilliant. I mean, that's great, Lynn. I mean, as you said, not medical advice for our viewers, but the advice is to work with your clinician in working out what's best for you and the treatment that you're on. So no, really valuable perspective. I mean, coming back to the booster, some questions are coming, you know, why 6 months after the 2nd dose? Why should it be sooner for those who are immunocompromised and maybe not had as good a response? Paul, Michelle, any thoughts on?
Interesting question. I mean, the timing of these vaccines, which are both new, of course, mRNA and Pfizer in the last year, 20th of November, 9th of November, I think it was last year, when these first results of these vaccines. The timing of optimal timing is still really being decided. America, of course, the Pfizer is 3 weeks. Israel is 3 weeks. Britain, we did 12 weeks. We made that decision to delay the boost. And actually, the evidence is accumulating. That's probably better. I think we'll probably get better protection from the 12 weeks, which is interesting. So that's 2. Even the spacing of the first 2 was uncertain. It would be unusual to give your 3rd vaccine very soon after that. And so I think we needed this 6 months to look at the relative waning of the antibody responses. I think it seems sensible to me, and it's being widely adopted around the world. Michelle, anything you'd want to add to that?
No, I think you're right. I think we've done, you know, the dosing interval change certainly seems to be promote better immune responses, and we needed data. I mean, or I suppose the JCVI needed data prior to making a decision about the doses. That's now come out, and it's a sort of scientific decision to continue. But so I think it's probably about right.
Brilliant. Thank you both. So changing tack slightly, we'll give you, Michelle and Paul, a bit of a rest. There's lots more questions I will come back to. But Lynn, just to come to you with a specific question, and just to say a lot of love for you and you sharing your story, being so open out there from the viewers this evening. So thank you again for sharing your story. But a specific question, picking up on something you said in your opener, was around the importance of disseminating research findings in the best way. And you kind of alluded to maybe that's not been done in the best way for some studies or in the UK. I mean, what would you like to see? How is there an example of best practice of dissemination, or what would you want to see from the various bodies and individuals in getting that information out to the general public?
So I might have a view in this, but this is not Lynn Laball saying this. So I was involved in another project with the Academy of Medical Sciences, which was on the winter pressures, and you know, the patient and carer reference group in that, and they also did some work with Ipsos Mori, and I think that everyone kind of feels that the communication wasn't as good as it could have been, and to me it's like, you know, research is done and then you get your publication, and then, well, you know, we'll do a lay summary and we put it out there. To me, it's been really thoughtful about who needs this information, who's got the most skin in the game. You know, who does it really matter to? And I think it really matters to patients as well. So, it's about being thoughtful of it. It's about working with patients to put information out in a way that they understand. And it's about working with communities, with charities. If there's some people that are especially affected, to go to them and say, right, these are our findings, how do you want that communicated? Does that make sense to you? And, I mean, I have to say all hats off to the BSI, because I think you've done that really well, Doug, and so is the COVID Immunology Consortium. And I don't want to seem critical, but it's sometimes just about a bit more thought, and that being important, not just as important as getting something in a high-ranking academic publication.
Great, thank you, Lynn. And I wasn't lining you up for a plug for the BSI there, but I mean, really good insights there, and definitely something we can all take away from that. I'm just looking through lots of questions coming through. So I mean, Jeremy, coming to you, you set the scene so incredibly well at the beginning. Of course, you've been at the centre of this, particularly in the UK, but also on global bodies. And there's some questions coming in around, well, how cautious should immunocompromised, immunosuppressed patients be, in particular during this autumn and winter as we enter a new phase of living with the virus, as you said, Jeremy? And, essentially, how do you see us getting through the winter and coming out the other end as good shape as possible?
Yeah, I'd just like to comment on what Lynn was just saying there as well. I mean, the other thing that's been transformational in the last 18 months is how the scientific community has shared information in a way that it's never done before. That's an opportunity as well to push it even further, as Lynn was suggesting, and I completely agree with. The scientific community of which Michelle and Paul are leading advocates of, have put the information into the public domain in a way that I think wouldn't have happened before. And I think we have to keep pushing on that door for the future. So I entirely support Lynn's view of that. On where we are at the moment in the UK, and again, Michelle and Lynn and Paul may have some comments here. Compared to September 2020, we're in a completely different position because we now have treatments, we have tests, we know much more about the virus, and of course we have vaccines. Positive. The negative is, in September 21, we're at a very high rate of infection in the community compared to 2020. And I think we can anticipate, and actually to give credit to the government, this was the message yesterday, I think, we need to be really cautious. We need to encourage people to have the vaccines. We need to push for people who may be concerned. Are they protected from the vaccines, from getting the maximum amount of information they can? Have we got antibodies? Have we got evidence that we might be protected? What can I do for shielding or seeing my loved ones or relatives? Because I think this is going to be, I'm afraid to say, a bumpy autumn and winter. About 1 in 75 people in the country are infected at the moment. That is very much higher than it was in September last year. And although vaccines have protected many people, as this call is all about, they may not protect everybody. And I think we are going to have to be very, very cautious. And the last thing I'd say, and I don't mean to be pessimistic here, but this infection is not going away. We're not going to come to the end of this pandemic in a sense. We'll move into a different phase, but I think we are going to have to put our mindset into thinking, what does the long-term future of how we as individuals and society will deal with this new human infection?
Fantastic. Really important perspective there, and definitely a word of caution. Great that we've got these vaccines, as you say, Jeremy, but there's still more that needs to be done to keep this virus in check. Coming back to some questions around immunocompromised people with weakened immune systems specifically. You mentioned, Michelle and Paul, about some prophylactic treatments and some of the studies that are happening. Is there something you can elaborate on there? Because there's lots of questions coming in about, well, what if these vaccines don't work or don't work as well for me? Is there something else that can be done? So Paul, Michelle, it'd be good to hear.
Yeah.
Do you want me to go?
Okay, Michelle, yeah.
Okay. There's certainly emerging evidence in terms of the use of passive immunity, so giving protective antibodies to those who don't have any antibodies, whether or not it be at the time where they become infected, so and become ill. So that was data was coming from the recovery study. And also there's prophylactic studies that have been coming out of America with Ronapreve, which is now approved in the UK, and a recent encouraging press release in terms of a long-acting antibody from AstraZeneca. So there are a number of different agents coming through which will hopefully provide an additional layer of protection for those patients who don't or are unable to mount a response to the vaccine.
Great.
Yes, I'd agree. It's as Jeremy says, incredible science again, and it's an old concept, injecting us with a preformed antibody from somebody else. It's probably some people on the line might have had it in pregnancy, rhesus disease, when you've anti-D, and what we have now is these antibodies which have been engineered. They last for months, maybe a year. So an intramuscular injection protects high levels of antibodies. It's super. And the data from the 20th of August, I think the American study came out, as Michelle said, really quite positive. So I'm looking forward to hopefully seeing those introduced for the most at-risk patients.
And I mean, on that, Paul and Michelle, maybe you've got some insights. Do you know what kind of timescale we're looking at for the introduction of these treatments for people who are immunosuppressed?
I personally don't know. No, I don't know. We've had some discussions, but I haven't heard any deadlines. I don't know if Jeremy or Michelle know any more.
No, I don't. I'm not sure.
I don't, but I do think here community groups, and as Paul said earlier, the charities in the UK, I would encourage everybody to push on that, particularly as we go into the autumn and the winter, through representatives, general practitioners, hospital doctors, and the rest of it.
Great call for advocacy there, Jeremy. Thank you. And some questions coming through from people who are primary immunodeficient patients. Have there been many studies looking at that community of patients, looking at vaccine responses and what can be done? Paul, Michelle, I know that's slightly outside of your direct area of research, but we have had quite a few questions coming in along those lines.
Primary immune deficiency, yes, yeah. There have been specialist research groups working in that area. We tend to think of primary immune deficiency, those patients are not making any antibodies. Some people are born not able to make antibodies and have antibody infusions. Obviously, in those situations, that going to have very poor antibody responses. We might be relying on cellular responses. Those are the groups that may benefit from these sort of prophylactic antibodies. I mean, yeah, I don't know a lot about that area. Michelle, do you know more on that?
No, I know that there are some research groups and they are in communication with the Octave team. So I know there is some research, but I haven't seen any data, I'm afraid, that I can report.
Okay, no, thank you, Michelle and Paul. And what we'll do, the BSI, we will try and pull that information together and include that on our website as well. So for those who are watching, there is some information on there, but we can definitely pull together some more of those, the outcomes of those studies for that important patient group. We've also got some questions coming in about what, the immunity antibody test score. What does that mean? What can patients take from that? Can they take anything from that around their immunity, either through natural infection or from vaccination? Paul, Michelle, and even Jeremy, if you have any thoughts, and Lynn, perhaps, from your communities with some questions coming in along those lines.
Yeah, we all want this correlative protection, this number that gives you protection, and it seems like a dream. We're not there yet. But of course, Jeremy and Michelle, I mean, in things like hepatitis B, we do that, don't we? We aim for a level of protection that we believe is a correlative protection. For COVID, we haven't got there yet. So, I mean, this was, there's been debate about whether patients and people should be looking at their antibody levels, because it's difficult to interpret what it means. It gives a certain level of confidence, perhaps, but we don't know the degree of clinical protection. I think that's really where we are. We're trying to work hard to define that. Michelle, anything you want to?
I think in terms of, I think from the general population, it's different from the immunosuppressed population, in my opinion. I think knowing whether or not there has been any response to a vaccine may be useful information. But again, we won't, even if you have an answer, you're not going to know whether or not you're protected. but certainly knowing that you haven't got a response may be useful, certainly as far as the monoclonal antibodies are concerned, because people without any antibodies may be in the front of the line for the monoclonal antibodies.
Great. Lynn, Jeremy, anything to add to that one?
No, I think that's pretty clear. Yeah.
Yeah, I think, sorry, I had a really quick look through the chat, and there's a great question that this is not just about hospitalizations and death, that this is about quality of life as well, you know, whether that's in terms of, you know, are people that are immunosuppressed more likely to maybe to go on and develop long COVID, and what are the, you know, the long-term consequences. And, you know, when I think of this need to know the number, and, you know, I had to stop myself sending off a blood sample to a private lab to get my number. And I think it's just all about understanding what this means for us. And just because the past 2 years has had such a profound effect on all of our quality of life, you know, and if we're coming out of this, it's like, who's coming out of this, and what does our quality of life look like going forward, either living with the disease or living with consequences of the disease. And that sometimes I think we need to think that this is a more nuanced picture than we make it, and things like long COVID and other consequences are. I know at the start of the pandemic, what I said to my husband was, I'm not concerned if I catch it and die. I'm concerned about my life being less than what it is.
No, such an important point. Thank you, Lynn, and thank you for doing my job for me as well and going through those questions. It's a very, very important question to pick up on. So I'm conscious of time, and I can't believe how quickly that 58 minutes has gone. But I've got two more quick questions, one for Lynn and one for Jeremy, and then a few seconds each. I'll come to each of you for any final comments before we wrap up. But Lynn, lots of questions around, you know, how have you got involved in research? I think there's a real desire for many of the viewers to try and get involved. So any quick tips on how people can find out how to get involved?
Make a real nuisance of yourself. Because I think it's when I started out that's what I did. I think so. I mean, one thing that you can do is have a look at the People in Research website, because people will advertise opportunities there. I think that there's some get involved with charities, and there's more opportunities now, I think, to get involved in research, either as a participant or as an involved person. But I think it's not, you know, it's funny because researchers say I can't find people, and then people that want to get involved in research says that I can't find opportunities. And I think we need a Tinder service or something like that. You know, where we swipe right or left or wherever it is. You know, I quite like this opportunity. Yeah. So I think we need to get better at that. Yeah.
Brilliant. Well, I don't think I've chaired a webinar where Tinder has come up before, so thank you, Lynn. And then final question, Jeremy. I mean, you mentioned in your opener that, you know, the phenomenal global effort from a research perspective, where researchers come together, sharing data, working in collaboration, and particularly in the UK that's close to us. That's been really beneficial. How do you think that will continue? Do you think how do we keep that momentum going forward, not just for COVID, but for other important disease areas as well, and try and really speed up the research effort?
Yeah, I think once you lift the lid on these sorts of things, you can't put it back. It's crucial we keep pushing for that to be true. The speed without cutting corners over the last 18 months has been extraordinary, and we must not lose that. It's brought treatments, it's brought vaccines, it's brought diagnostic testing to patient benefit quicker than has ever happened really in history, and we mustn't let that go back in the box, if you like. But the other thing is there is still a huge amount of research that needs to get done. There is enormous uncertainty still, I'm afraid, about a lot of the issues that Lynn has talked about. And I'm a volunteer in a number of COVID studies, and I cannot just overstate how important it is that we still keep asking questions, because there are still a lot of unanswered questions out there.
Fantastic. Thank you, Jeremy. So final comments from each of you before we wrap up. I'll do it in random order. Michelle, let's come to you first. Any final words of wisdom for our viewers?
No, just I said I think that having access to research for all patients is incredibly important. And actually what's happened in COVID is collaboration amongst specialties. So never before have we worked with rheumatologists, with cancer specialists, etc. And there's so much overlap. So it's been good for that reason.
Thank you, Michelle. Paul.
Yeah, just one thing, patient involvement in research. I mean, great to Lynn just to say it right at the start to keep us real and to make sure we do it. So, I ran the CIC, Lynn was on that, 10 patient in groups, been transformational. And when we ran that CLL vaccine study that I talked about, we got a patient group and we meet with them, they guide, they read our literature. I'm ashamed to say this, but it might be true. Maybe a year or two ago, we might not have even had that. That was wrong. But now we absolutely realize we must. And that's been a huge change that we will never go back on how we involve patients. And even this virtual conference, this tonight, I mean, many of you used to travel. Michelle and I used to go to conferences in an American hotel. Jeremy would be on a helicopter to Ebola in the Democratic Republic of Congo. But, now we're all on Zoom. But the idea of talking to patients groups on Zoom is fantastic, and we can reach 500 people. That won't go away as well. So PPI, patient public involvement, is a major legacy.
Fantastic, thank you, Paul. Jeremy, any final thoughts?
Yeah, the thought I would have is the information is changing so quickly that I think we should push Lynn and Michelle and Paul, Doug, for you to repeat this just before the end of the year. An update on anything for new information that's coming, because there will be a lot more information coming in the next 3 months. And, I would be very happy to join. I've learned a lot tonight to go back in 3 or 4 or 5 months' time and just repeat this exercise if it was of use and of help.
That's brilliant, Jeremy. We'll definitely do that. And diary notes will go out following this meeting so that we book you in. So thank you, Jeremy. And last word to you, Lynn.
So I would, I think it's great what's happening in the space of patient public involvement, and I would say that it's about honest conversations, and it's about relationships, and it's about collaboration. And when it works, it's the best feeling in the world. So thank you. And maybe the last word should be to the Health Research Authority, who said that patient public involvement was important, expected, and necessary. And I don't think I can do any better than that.
Brilliant. Great words of wisdom. Well, that does bring us to the end of this webinar. I'm told there's lots of thank yous coming in on the chat at the moment. So really great stuff. I mean, thank you so much to all of our speakers, to Lynn, Jeremy, Paul and Michelle. Really appreciate the time that you put aside to come and join us this evening, and the insights and the opinions that you brought with you. Been absolutely fantastic. Loads of positive feedback from the viewers. So thank you, speakers. Also, thank you to all of you who have been viewing. There's still hundreds of you here listening and watching, which is great, even though we've run over by a few minutes, which I am sorry about, but really great to have you here this evening. The good news is this has been recorded and it will be made available free on the British Society for Immunology's website. So if you want to see it again, it will be there. But also if you want to tell your friends, your family and others about it, then please do feel free to cut and paste the link and send it over to them. So give us a few days to get it up on the website, but it will be there, and we'll send an email out to let you know when it is. And of course, while you're on the BSI website, have a click around the resources that are on there. There was a link that went up earlier, lots of information around coronavirus, around COVID and around vaccination on there. And if there are some gaps, then just send us an email and we'll get some experts to work on that and pull that information together. So that just leaves me again to say thank you, speakers. Thank you, everyone who's joined. Been an absolute pleasure chairing you this evening. And we will see you again before the end of the year, apparently. Thank you, Jeremy. So thanks, everyone. Have a good evening.